Let's move into module 2.1. So to begin this module, we're going to talk a little bit about the fertilization to give you an understanding that inside the ovule, and once that egg is fertilized, that's where the genetic recombination happens. And so we don't really have a lot of control over that. We're really just trying to find the phenotype that suits us, but we don't really have an idea of getting the exact phenotype. The genotype, right? Remember from the question in the exam that we didn't really know what the genotype was, and if we did, then we could have a better plant breeding program. So understanding what needs to happen in order for that genetic recombination to occur is important for understanding where on the chromosomes that we're going to start trying to modify those genes. And so there are several objectives in this. We're going to talk a little bit about fertilization. We'll talk about ploidy and some of the crop breeding objectives. You can go through these and look and see just what the importance of this is. The double fertilization is going to be important as far as for seed production goes if you ever get in a seed breeding program. A little bit about ploidy. Ploidy is important because you need to understand how many sets of chromosomes so that you can match those chromosomes up, right? 2 with 2, 4 with 4. But if you had 2 with 4, then you wouldn't get 2 or 4. When we get into the ploidy, you'll understand what I mean there. I'm going to briefly introduce some of the traditional breeding methods that Mendel used, that Norman Borlaug used, in the 40s and the 50s when he was trying to develop some of the hybrid vigor in our crops. And then just a recap, some of you may have had me for plant science, some of you may not have, but just to go over a little bit about the foundation of genetic modification, genetic engineering, the way that they did it back in the late 1990s, or the late 1980s, early 1990s, when they were just beginning this new technology. This is going to help you understand how far we've made progress from our traditional breeding, the general genetic modification, and then now some of the new tools that we have. And I want you to keep in mind that this is all about finding the genotype. If we know what the genotype is, we can either A, select for it, or B, we can modify it. And that is the most important part of Module 2, is understanding something like CRISPR-Cas9 or QTL, so that we can actually map a genome, know what genes affect what traits, and then modify those genes quickly, efficiently, so that we can get new crops to the producers without taking 8 years. So again, the key takeaways, we're going to talk about fertilization, ploidy, the old way of traditional breeding, and I don't want to say the old way of genetic engineering, but probably the foundation of genetic engineering is probably a better term for that. So let's get on with embryogenesis. We remember from plant science that there are a male part of the flower and a female part of the flower. We have the anther and the filament that make up the stamen. This is where the pollen grain is. That is going to be the male gamete, the pollen grain. And then we have the female part, which is at least the carpel, which is comprised of the stigma, the style, and the ovary. And so the female gamete is actually the ovule. So let's run through this life cycle. We have 2 initial processes. We've got sporogenesis, and this is the pollen. Think about the size of a pollen grain. The actual pollen grain is microscopic. It's no different than human sperm. We need to see this. We can't see it unless it's under a microscope. And then megasporogenesis is the ovule. So how big would you expect a pollen grain to actually become? Not very big, right? How big would you expect something out of the ovule to come? And consider that in the ovule, we are going to fertilize an egg that becomes a seed. The pollen grain does not change in size. The fertilized egg will change in size as it makes the seed and the fruit that protects that. So we've got pollen from the anther that falls on the stigma, moves down the pollen tube in the ovary and the ovule, and fertilizes this egg. That egg then becomes the fruit and the seed. That seed is then dispersed. We have germination, and we continue that life cycle. So a bigger look at that, again, pollen from the anther onto the stigma, down the pollen tube, fertilizes the egg. We have a zygote that turns into a seed, that turns into fruit. The seed is then dispersed, germinates, and continues that life cycle. It is in here where we get that genetic recombination. So for our microsporogenesis, it's kind of important to understand that if you think about a pollen grain, again, remember it's microscopic, but at the microscopic and the cellular level, that it's kind of like a jawbreaker. There are different protective coats around this in order to protect the genetics, in order to protect the cells in the nucleus, the generative nucleus and the generative cell, because that's the most important part of its survival. And so the microsporogenesis is important for the creation of, it is the creation of the male gamete, and then the megasporogenesis. And so again, when we think about mothers, we think about that they are multiplying, and there's this weird thing that when cells divide, they're actually, or cells multiply by dividing. So we have megasporogenesis in that we have a 2N that forms a 4N, that forms 4 megaspores, each with 1 N. And this N is diploidy. This is the set of chromosomes. And so if we look back here, we see that the sperm from the pollen is 1N. The egg is 1N, and that forms 2N. So N is the number of chromosomes, and that could change. Then, the sets of chromosomes. All right, so we have this megaspore that undergoes meiosis. This is just regular cell division that forms 4 1N or haploid cells that can then be fertilized. There are some more mitotic divisions. I'm sorry, this is meiosis. This is asexual. Not asexual. This is a reproductive This is a reproductive regeneration or generation of that. Mitosis is just general cell division. Okay? Ultimately, at the end, we're going to have 3 nuclei, and then 1 diploid. And again, we're not going to get so much into this where you have to go through and explain this. I want you to understand there are processes and there are things that need to happen in order for that genetic recombination. And we don't have control of that. We only have control of altering the DNA. All right, inside the ovule, we have the integuments. So this is a protective coating around this nucleus that kind of like a womb that protects this megaspore. Next, we have the micropyle. the micropyle, it's the opening, and then the embryo sac. So we have an egg cell, 2 centrigids, 3 antipodals, and 2 polar nuclei. Again, I just want you to understand that there are different cells inside of the ovule that all play a role in the genetic recombination. All right, went back through this again. We got the pollen grain on the stigma, grows down the pollen tube that comes in through the micropyle that fertilizes the egg. And that generative nucleus divides into the 2 male gametes and move into the tip of the pollen tube, right? So it grows down. And also something to understand in this, that the plant knows if this pollen is from its own cells or if it's from a different cell. And each pollen grain is shaped a little bit different. There are different chemicals in there that the plant can sense and determine if, let's say this was a cross-pollinated plant and it's a complete perfect flower in that the pollen from this plant falls on this stigma. As the same pollen falls on this stigma and moves through here, the plant will start to notice that this isn't the right pollen type in order to fertilize this egg. And so it will take measures and it will dissolve or disintegrate that so that it doesn't risk fertilizing an egg for no good purpose. All right, just a few more pictures, a little more in depth to kind of see what's actually going on here. And if you get into a plant breeding class or you happen to follow on and continue on into genetics, this would probably be like one of the fundamental things that you encounter when you're learning about plant reproduction. Maybe in botany, you might go over double fertilization a little bit more. In plant science, I don't really go too in depth with this because it's a freshman level course, and that if you do move this route, you'll get plenty of opportunities to interact or engage with that. So, in flowering plants, we have 2 sets of fertilization, the sperm nucleus and the egg cell. If it fertilizes that egg, then the centrigids will disintegrate. So they're kind of like the protectors. And until the right pollen or the right sperm nucleus gets into that ovule, they're there to protect that. And so if we have successful conception, so to speak, the centrigids will disintegrate. If we fail to fertilize that egg, the centrigids will then assume the role of that egg cell. They will change and become, or at least gain the ability to be fertilized. And if you really want to know more about that, here's a YouTube video that explains a little bit about that genetic recombination. Probably just something good just to look at, just to improve your knowledge and maybe hearing it from a different source. So why, Doc? Well, why is this important? It's the basic process for genetic recombination. And so we have to understand self-pollination and cross-pollination in order to develop hybrids and lines. So if you go into seed breeding, you're going to need to understand a little bit about this so that you can continue to increase that population and keep that genetic consistency. All right, something that is important, and I'm going to definitely readdress this in the lab when we talk about the dihybrid crosses, because we have ploidy, which is the number of sets of chromosomes, and humans have 26 or 46 chromosomes, or 23 pairs. Therefore, we have 2 sets, and we are diploid. 2N. Notice that the N can change, but that there are two sets of how many ever chromosomes this particular organism has. And so remember that on these chromosomes we have alleles and we have homologous and we've got homologous alleles, we have heterozygous alleles, and we don't know which alleles are going to cross, and we hope that we get big A, but it's possible that we get big A, little a. If you increase the number of chromosomes or the number of sets, then you increase the randomness of the genetic recombination, because there are more opportunities for these different alleles and genes that turn certain characteristics on that we're looking to improve. We're guessing, and so if you thought the dihybrid cross with a 2N, a diploid plant was challenging, big A, little a, big A, what if you had a tetraploid plant that had 4 of these different chromosomes? So as we increase the ploidy, we're going to increase the difficulty of the outcomes that we're looking for in our breeding program. So just to look at some of the polyploid plants, something like wheat, tobacco, banana is important. I'll talk about these in a minute, and then I also want to talk, at least notice the boysenberry. So let's take a look at this. If you recall, humans have 46 chromosomes, but 2 sets, which means each cell has 23 chromosomes in it. Remember, 1N plus 1N equals 2N. So if we have 6N with 42 chromosomes, how many chromosome sets are there in this? 42 divided by 6 is 7. So there are 7 sets of chromosomes. 48 divided by 4, N equals 12. Correct. It's kind of like genetics meets algebra. And so understanding how many sets of chromosomes that you have to deal with is going to influence how many different, the more chromosomes that you have, the more extensive it is. The human genome is a very complex system because we have 23 chromosomes. Well, that's different from 7. It's much easier to breed wheat than it is to breed humans who have more chromosomes to add genetic variability to that. And then making sure that all of those line up and that you get exactly the right ones. But why is this important? Why would we want to increase the ploidy of our crops? Well, we're going to increase some vigor and we're going to get some enlarged crops, and we'll see this in just a moment. We can also breed for seedless fruit, and this is, many of us get seedless watermelon from the store, so that's one advantage. We can overcome some barriers in hybridization. It's important for forage breeding, and then also some disease resistance. And understanding the ploidy kind of gives you an insight as to how difficult it will be to achieve this goal that you're looking to achieve. So some of the applications, we can do mutation breeding. We can do seedless fruits, as I mentioned, forage breeding, disease resistance, industrial applications. When we change the ploidy of this, we are actually increasing the size of the plant, and we're also increasing genetic material, vegetative material. If it's something like corn, what if we just had massive corn plants or corn cobs that we were using for ethanol? the bigger the plant, the less the less number we would need, and therefore the increase in efficiency. So let's take a look at diploid and tetraploid grapes. Notice how small these are. These are our standard type grapes. We have now doubled the size. And I really like this picture of these strawberries. And I had not considered this. I just thought, man, they just grew some really big strawberries. But it turns out that these are actually 4N or tetraploid strawberries. Notice how much bigger they are than our traditional strawberry. Notice some of the changes in the morphology of that fruit. And do we care about that? No, I don't care about this. I'm cutting that top off and eating a very big strawberry, and it is relatively good, depending on how you are about store-bought produce versus maybe something that's grown fresh from a farm. All right, seedless banana. And many of us have probably never experienced this, but previously, and I don't know how long ago this was, but bananas used to have seeds in them. And now we don't have that. Because who wants to pick through this? Much like our seedless watermelon, who wants to pick through the seeds? They're kind of a nuisance. So, over time, they figured out how to breed seedless watermelon. And the way that they did that, and so we're going to do some math here, it's just very simple. The way that they did that is they took a diploid watermelon, 2N, and they crossed it with a tetraploid watermelon. Now this is challenging, and so you're going to need some extra chemicals in order to do this. Not that this dinitroaniline is important currently, but if you get into a breeding program, you might hear of things like dinitroaniline or I'd have to. It's slipping my brain. Colchicine. Okay. Anyhow, we've got this chemical that we need to add to this in order to cross a 4 with a 2. Well, when you make 4 plus 2 and you divide that, that is now 3N. And either one of these, like you're going to have an extra set right here, this extra set of chromosomes that doesn't have a partner, so to speak. There's no counterpart. And so it doesn't make seed. And it's sterile. Because if it doesn't make seed, then how is it going to reproduce? That's a really good question, isn't it? How do seedless watermelon reproduce? They're bred that way. They're bred that way. All right, let's take a look at Arabidopsis as we start to increase our ploidy. Okay, we've got 2N. This is a very small plant. Notice that we've doubled the size, like we've doubled the ploidy, so now we have 4N. Notice how much bigger it is. Now we've increased that we've doubled it, we've tripled it, and notice how much fuller the leaves are, so we don't have the space here. And then finally we've 4x'd this, and now you can see that there is no space. Those leaves are big. We have a very full plant. So we've increased the biomass of this Arabidopsis by increasing the number of chromosomes. So we're going to increase the size. We're going to increase the vigor. We can have sterile triploids. And we're mostly using this because seeds are kind of a nuisance in our consumption. Overcoming barriers and hybridization is important because remember, we want to have genetic variability. And if we've already bred through enough plants, how do we get the genetic variability if we've already, we've already expended any of the genetic variability. We've done all the crosses that we can. And so one way to do this is we get interspecies hybridization, in that we find something that is in the same genus and we bring that in and cross it. And we can get some of that vigor out of that wild species, the wild type, and introduce it into our more cultivated. And then also pest resistance and stress tolerance. Let's look at this one. Do you really think that this plant has a lot of defense against any sort of environmental or any sort of insect? No, they'll eat this up quick. There's no protection here. Look at how light yellow and green it is. Is that growing very fast? Do you think this one has an opportunity to outgrow attack for pests or conduct photosynthesis? Which one do you think conducts photosynthesis better? The 4N. And so then we can move that over to the 6 and the 8. So we can increase the ploidy in order to increase resistance or function. Ooh. Some of the drawbacks is that they can be undesirable or unstable. And so when we talk about undesirable or unstable, is that we might only have these for just a little while before the variability, the genetic recombination doesn't work as well. One big problem is sterility, as we talked about with the seedless watermelon. How do you breed? I mean, how do you breed seed for seedless watermelon? And then on the inheritance and population genetics. So, moving into breeding, we've got breeding programs, and it takes years to develop this. We'll see this in a moment. If you've had me for plant science, if you recall, we left out at the end of the class talking about breeding objectives, knowing that at some point in time as a freshman, GMOs might be a little much to comprehend right there. But now that as you're juniors and seniors and you're in an upper division 4,000 level course, you can start to see that there's a lot more to it. And so now you've had exposure to some different aspects of agriculture, and you can finally grasp and see bigger pictures and start to see where this is kind of intertwined there. So it used to be that in the late 1800s, early 1900s, it took about 1/10 of an acre let me do the math on that. What is that? 1452 divided by 43.5. It took about 0.03 acres to grow 1 bushel of corn. As we so somewhere here in the 1950s, we get Norman Borlaug, and we start changing some of the plant morphology and the characteristics, some of the nutrient uptake. Here we have genetically modified organisms. Somewhere in the 90s, we're starting to improve that. 2009, and so somewhere between 2009 and 2030, which is coming up faster than we think. Now we're at 145.2 divided by 43,560. Now it takes us like 1/100th of the size. 1/10 of the size. It used to take 1452. Now it takes 145.2. So we've increased the amount that we can grow on the size. And so that improves our efficiency. That improves how much we can actually produce with the land. We don't need as much land in order to produce enough. That is going to be debatable, what is enough. And then also, how does excessive cultivation of this crop affect our soil? Some other things to consider. And so plant breeding is a reason why we have the variation in our plant food options. Everyone wants to come up with something new. Cherry tomatoes, or we want green tomatoes, or we want, look at all the different types of tomatoes that we have. Look at the different onions that we have. maybe even corn. We've got sweet corn, we've got regular corn. So we are trying to manipulate the genetics in order to provide something that we find desirable. So our plant breeding objectives, and I just got a big star by this because you're likely to see this again. So we need resistance. Resistance to insects, resistance to diseases, just environment. What about drought? I noticed some of you mentioned about drought. We've got resistance to, or at least adaptation to wet conditions. If you're considering animals, what about feeding quality? Not only for animals, but also for humans. What is the quality of the product? Market quality. How good does it look? Let's think about apples. Is it this very deep, like beautiful, glowing red, or is it this dull sort of, there's a couple of blemishes on it, or tomatoes? What are the size of it? Seed quality. Seed quality is going to be something that's important because that's what we're going to plant, right? Growth, habit. This should actually be growth habit. So growth, is it tall? Is it small? How does that, does it grow wide? Can we grow it in a very narrow space? We got market quality. I don't know why that's in there twice. We'll see in just a moment. And then productive capacity. And this is kind of, when we talk about productive capacity, this is yield per se. It is very difficult to specifically breed for increased yield. You breed for increased nutrient efficiency, increased biomass, and you hope that that leads to larger corn ears. I'm just going to use corn as the example. All right. So we got resistance, and disease and herbicide resistance is becoming important. One of the problems with this is that our insects are now becoming resistant to our insect resistant crops. And so now we're faced with another dilemma in that we've created something that is not what we created it for or to do. To be able to handle environmental conditions, and this one's going to be huge. I noticed a lot of students mention drought tolerance. We're going to have cold tolerance. In Tennessee, if you've heard me talk about climate encroachment, we're going to need increased heat tolerance. In desert areas where there's not a lot of rainfall, or the rainfall or the water table has sodium or calcium in it, we're going to get alkalinity or soil salinity. So how do we breed for these things? Next, adaptation. So variable photo periods. And one thing that I know they're working on at Auburn is cover crops that are actually flowering earlier based on photo period. And we're using this because we expect to either have shorter or longer growing seasons for that. Maybe they need to adapt to heavy grazing or frequent cutting. In Mississippi, we cut Bermuda grass probably every month. Every 3 to 4 weeks, it's hay season for us. In Tennessee, it's not, because we grow tall fescue. Tall fescue doesn't necessarily handle our heat very well, so our heat tolerance is low. So having a heat tolerant forage species is going to be beneficial that can handle heavy grazing or frequent cutting. So we're changing the growth habit to increase yield. Feeding quality for grazing. Is it palatable? Is it easy to chew? Does it have a lot of lignin in it? How does that interact inside of the cow's stomach as it's trying to process this? What is the nutritive value? And this one's going to be important, I believe. Nutritive value is going to be important, and I've probably mentioned this before, in that we need more nutrient-dense food or nutrient-dense substrate, biomass, so that we don't need as much of it. So it's nutrient-dense. Market quality. Again, this goes to nutrient density. If we can increase proteins or starches, better processing, better color. And you may have heard me talk about golden rice. And when we when we get into module 3, we'll look over at some, the impact that golden rice actually had on populations in Africa who were suffering from vitamin A deficiency. Seed quality. So seed setting. Are we getting more seed? Are we getting quality seed? Can we store it longer? So let's think back to our, storing our germplasm. Can we store these seed for a long period of time, 20 years, and when we pull them back out of those chambers, it performs just like it did when we put it in there? Are they a larger size? What is the dormancy and germination? My major professor is a plant breeder for switchgrass, and what he worked on was speed of germination for switchgrass. And I'm going to bring that up when we talk about mass selection and how we determine that. Growth habit. So Norman Borlaug, father of the Green Revolution, in the 1950s, they had issues with wheat lodging, and this could have been from over-fertilization, in that they were over-applying nitrogen, trying to increase the yield, and what they did is they forced that plant to grow very tall without growing very wide. And so this shoot-to-root ratio can be important. In that, remember I talk about that the taller the plant, the more roots that you need to grow. And so also the stalk. So if you have a tall plant with a thin stalk and little roots, is that going to stand upright? And what happens when you drive a combine through here? Well, you miss all of this. And so now you're driving the combine over an area. So not only did you lose the crop, but you're also technically losing the efficiency of driving that combine through the field. And then harvesting quality. So we can have wheat in that there are these awns, and when you pick those, they need to be processed so we can reduce the awns and the fuzz so that they can be processed quicker and turned into, let's say, bread. And then finally, productive capacity. And so this is going to be greater vigor, better fertility, faster recovery, and again, I call this yield per se, in that we are going to impact plant growth in nutrient use efficiency in order to influence the nutrient density. Okay, traditional crop breeding methods. Originally, there are two. We have mass selection and then hybridization. And so I mentioned that my major professor was screening for germination. He was trying to get faster germinating seed from these different plants. And what we did is, we just got a bag of seed of switchgrass and took a seed lot, like a bunch of seed, let's say 5 pounds of seed, and germinated them for X number of days. And if a seed germinated under 7 days, then we moved that seed forward. If it germinated on day 8 or 9, maybe it made it. But if it took 14 days, then we don't want that. So we actually selected for the ones that were tolerant to our screening. If we're looking for drought tolerant seed, you can do the same thing in that you try and germinate that seed in low moisture conditions. What about heat tolerance? You germinate that seed in high heat conditions. What about, we did one for salinity. How do you select for salinity? We took a lot of seed and germinated it in various levels of saline solution. Those that germinated were moved forward. We kept the desired survivors and bulked that progeny. Next, we get into the hybridization, because now we want to import this trait that we mass selected for into the offspring and make sure that that is continued. So we combine this with another plant maybe, and that we grow those, we get those seed, we test those seed that came from this F1 generation and the F2 into our screening. So drought, heat, saline, and continued bulking that progeny through cycles, such that ultimately we found that, you know, we have a 90% germination, or a population of seed that 95% exhibit this phenotype that we're looking for, and that phenotype being this resistance to whatever factor. If not, if we found that this plant exhibited qualities A, this could be our yellow seed, and this could be our round seed, and then we have to cross these, we get a phenotype, then we self this and we get the F2 with different phenotypes. And so I asked the question about the dihybrid cross because I want you to think about how long it would take in order to get the yellow wrinkled and, let's say, the green wrinkled out of this population. And when we look at the dihybrid cross in the lab, we'll be able to see why this method takes so long. This takes like 8 years in order to get this. We had 9 cycles of this, so 9 growing seasons for this mass selection to occur. All right, so our hybrid techniques, we're going to have our inbred lines. We'll do the test cross and the production crossing. Once we get enough of those seed that have our desired characteristics, typically what we'll do is we'll send that off to a seed producer who will bulk that. And when they bulk that, we can continue making more of these seed to be sold for value. All right, some of the advantages are that didn't take these out. I'll have to get you this slide right here. I'll send this in an email. But we want uniformity. I think like this one is uniformity. We want all of our plants to germinate at the same time. We want them all to reach the same growth stage at the same time. We want them all to flower at the same time because we're going to need the pollination. And we also want them to reach physiological maturity at the same time. I can't remember these two right off the top of my head. Some of the disadvantages. It can be more expensive than open pollinated. We really can't replant that grain without having some yield reduction. And then this one's important here. This is the inbreeding depression. And it is the manifestation of deleterious alleles. And so when I say deleterious alleles, I mean the ones that are not advantageous. And I'm going to say deleterious alleles. Let's think of insect or disease resistance. If the little r is not resistant to a pathogen, why would you want to continue having that in your population? And you can't, if you're just doing hybridization, you're going to get some hybrid vigor for a little bit, but eventually if you continue to do this and you don't get any genetic variability coming in here, eventually your population will get stunted and you will lose all of this pretty much hybrid vigor, everything you worked for. If you don't bring in new genetic material that is going to continue, then eventually your population will decline. All right, so we continue to self these and we don't have big Y, big Y, big R, big R, we can get inbreeding depression. So we'll look at the timeline. A line development takes, about 10 years. Then we've got to test that to make sure that it works well in the field. And then it's about 1 or 3 years in the farmer's field. And it only lasts for, 2 or 3 years before we start getting the inbreeding depression or we start losing some of that hybrid vigor. The farmer now has to get a new variety that's been in the chain for 5 years, and it might be that we don't have enough new seed coming down the line, or we have to bulk that seed. There can be a lot of issues with this here. When we do speed breeding, where we're actually intentionally changing and genetically modifying, we can get, this takes what, 10, 13 years? We're now getting the seed to the farmer, like the new variety to the farmer, in under 8 years. So a big change in the timeline so that we can meet these challenges. And as our climate changes, we're going to need to be even faster and faster at that. We just don't have the time to go through this process. We just can't do it anymore because the climate is changing faster than we can keep up with. Our plants are starting to suffer from drought, heat, salt, all of these environmental factors, and we need to get a solution soon. And so the purpose of this class, at least Module 2.2 and Module 3, is to get you introduced or at least be aware of some of the techniques that sort of outdate this and allow us to overcome these challenges sooner. All right, so just another look at how we used to do this. You know, conventional plant breeding, somewhere between 8 to 10 years. We've got different back crosses that we need to do in order to get some of those deleterious alleles out. So this back cross is what I was asking about in the dihybrid question on the exam. We didn't talk about it, but your responses were that you needed to figure out how to get the little r's out of the genotype. And the way that we do that is with back crossing. We can do mutation breeding. Obviously, this is chemicals and radiation. I don't think any of us are really signing up to work for a company that's doing chemical or radiation mutation breeding. Just not us. Transgenic breeding. This was Roundup Ready soybean, Roundup Ready corn, Roundup Ready cotton, in that we could change the genetics. We finally figured out a way to change the genetics, and we can now get a plant, a new variety, to the producer in about 4 to 6 years. And now with our genome editing, CRISPR-Cas9, quantitative trait loci, we can get that also in about 4 to 6 years with specific. We can so this is general in that we're changing one specific thing. When we have genome editing, we're actually going in there and changing multiple things, and we know what is being affected by each gene. So this is like genome mapping. Okay, moving into genetically modified organisms. Some important things to understand are like genetically modified. It can also be genetically engineered. You might hear these terms used separately, but they're really kind of the same. They all sort of fall under the same umbrella. So we have transgenic, trans meaning cross, genic meaning genes, and that comes from another species. Next we have cisgenic, cis meaning same, genic meaning genes, and comes from the same species. Some of the things that we're using, at least genetic engineering, is trying to figure out recombinant DNA, human growth hormone, cancer treatments, vaccine productions. Like, this is important for continued human quality of life, so to speak. Depending on how you see that, I don't really want to get into some of the problems or some of the negative impacts that we might consider with vaccines. Previously, maybe in my generation, these were very important. So why are we doing this? Nutritional enhancement, for one. I talked about vitamin A and golden rice. Rice does not generally produce vitamin A, but it was a staple crop in that African country where those children were suffering from vitamin A deficiency. They figured out a way to go in and genetically modify a gene to produce vitamin A. It normally wouldn't do that. They changed some things around with the DNA so that the proteins started making vitamin A. We've got stress tolerance, we've got disease resistance, and then biofuels. And so we are now going in and modifying these plants to, let's say, produce less lignin, because lignin is an issue when trying to convert that to ethanol. So we don't need years of breeding, and if you can find the gene in any species and you believe that it's going to work, well, let's give it a try and let's see what happens. Some of the drawbacks: you don't really have any control over where the gene goes, at least in the previous methods of genetic modification. In the newer ones, in the more novel techniques, CRISPR-Cas9, QTL, we actually do have control over that. We are specifically forcing or specifically directing that Cas9 protein to a specific gene that we can knock out. So overall, we're going to identify that organism and extract that DNA because it has something that we want. We're going to clone that gene. We're going to modify the gene to work better inside the recipient organism. I want you to think about this like an organ transplant. There are some things that have to line up. We just can't take DNA out of a gorilla and stick it in fish. So like there needs to be some research done on how this can actually work. How are they compatible? And then how do we deliver that gene to the recipient? So we identified it. We have this gene delivery. We're likely going to infect that cell. If you had me for crop production, we talked about like a virus. And once that virus gets inside of the cell and those cells replicate, you then now have that virus or that new DNA inside of every cell. And then finally, we have the expression of that. We have quick expression, and we can test very quickly on whether or not we've achieved our mission. So it used to be that it was micro-injection, in that we had this very small needle that we are, micropipette, that we're actually injecting that into the cell. You can imagine how tedious this is, and then also how expensive that is. Next, we have particle gun biolistics, in that we used to put this genetic information on little pieces of tungsten or gold. That's probably expensive as well. And we have this DNA that we basically shoot through the plant, and we would expect that when that penetrates, that we are kind of getting some residual inside of that plant, and that DNA is then transferred into the cells. And then finally, we actually have a plasmid where we are injecting, we're actually infecting a plant with this bacteria or this virus that is going to impact or influence the DNA. And so we have our TI plasmid. And again, I don't really want to go over like exactly how they do this, but I do want you to understand that there's a plasmid. We have an infection. We're going to have this crown gall. Once this gall infects this plant, you can see that we're starting to have this change, and we have now started to infect the DNA of this plant. We can also do this in vitro, where we're doing this in cultured cells, and then we grow those in a beaker under in a growth chamber, where we're specifically making these new cells into new plants. And you can just imagine how long it takes to actually grow a cell into a plant. How much energy that requires, how much cost of the space and the chemicals and everything that you need in order to do this. This is a very tedious process. It takes a very long time to do. Not as long as back crossing, but it still takes time. One of the biggest things that at least, began or, revolutionized our crop production was glyphosate resistance, and that there is this shikimate pathway inside of the plant that was not able to uh, metabolize the glyphosate molecule. So they found an organism that had a shikimate pathway and tested that organism for glyphosate resistance. And when they found that modified shikimate pathway was able to metabolize the glyphosate, they said, hey, let's try and put that gene into a plant. They have the TI plasmid. They either injected, this one they actually was probably the crown gall, grew that plant from a cell culture into a plant, then they tested it, they sprayed it with glyphosate. If it died, well, then it didn't work. If it lived, then that means that it worked. So we actually wanted to keep the ones that were alive and get rid of the rest of them. So we talked about at least these two, the traditional breeding, the mutagenesis, RNA interference. That's way above, that's probably way above my pay grade, my skill set. I'm aware of how we are switching off these selected genes. That'll be module 2.2. And then we just kind of went over our transgenics in that we are specifically putting those genes where in a known location. We know the gene that we need to select for, and we're altering the gene. Here, we're actually going in, this is going to be something like CRISPR-Cas9, where we go in, we target a gene, and we either turn it on or turn it off. And so here's a timeline of our genetic crops. Some of the crops that we have, we've got corn, we've got soybean in the 90s. We've got some squash and some soybeans, papayas, canola, just a few of these crops that are staples. We do have these genetically modified. A look at more some of the traits, or at least some of the crops that we are modifying. Some of this, maybe you eat some of these yourself. And why we've done this. And it kind of makes sense in that, for field corn, we need to have insect resistance or herbicide tolerance or drought tolerance. Those make sense. So we need to make sure that we understand what we're actually using these for, because you're going to have people that are going to be anti-GMO, that are going to be anti-agriculture, and they're just going to tell you what they saw on TikTok. Those of us who know that we're doing this because it makes our life easier to meet the needs, or at least the food security of those around us. Just another look at some of the statistics for our soybeans. And a lot of these crops that we're actually growing and using. Let's take a look at this alfalfa. We've got less lignin, which means greater digestibility, which means greater nutrient uptake for the animal. And if we have greater nutrient uptake, we get increased muscle production, less fat. And what are we buying at the store? We're buying the animal muscle with less fat on it. Some of the other ones, we have now salmon. And so they're growing this salmon to grow a little bit bigger. And so they've found some genes that influence how fast these grow, whether that is good or not. Not here to debate that, just to understand that these are already in our system and they're going to be very difficult to remove. All right, almost done. This one's an important one. This is GMO and crops. And so if you're in my crop production, you'll probably see this again in Module 3 because it's important on how we can use genetic modification for our plants to combat insects. So, corn earworm, very bad for our corn and our cotton, our boll weevil. Bacillus thuringiensis is a bacteria. We took that TI plasmid, we infused that into the DNA so that these genes actually produce this Bt toxin. They produce this Bacillus thuringiensis inside of the plant. And when the insect eats those Bt crystals, there are toxins that bind to specific receptors in the gut. You can see these here. So it attaches in certain places that stop that insect from eating and break down that gut wall. And so once that gut wall breaks down, we can now have the gut bacteria proliferate, and it essentially starves itself. Like it can't eat. And if it can't eat, it can't get bigger. And if it can't get bigger, it dies because it's also being eaten from the inside out. So this one is important to at least have an idea to understand how, just a basic general knowledge base of how Bacillus thuringiensis impacts our corn earworms. And if we don't have to spray insecticides, which they're trying to get us to do, is to spray less insecticides, but we can't use, or there's a big uproar about genetic modification, then what do they expect us to do? One issue, and we are seeing this now, and we're having to change some things and come up with new solutions, is that we now have insect resistance. We've fed this so much, the Bacillus thuringiensis, that the corn earworms have adapted. We also have the same thing with our herbicides. We've sprayed so much Roundup that pretty much everything's resistant to it. And yet producers are still spraying it. All right, so here's a look. We have our Bt corn on the left. Looks good. I'm going to buy this in the store. And you probably don't see this in the store because everything's got husks on it, right? Well, how would you like to buy corn at a farmer's market or in Walmart and get home and go to put everything on the grill, and this is what you open it up to? And you see these little worms crawling around all up in your food. How do you sell this? How do you spray for this? How do you spray for this insect when it's housed inside of the corn husk? What, do you go around and put a shot in each corn ear? Each cob? No. So this particular application makes sense. For our cotton, we have these insects that are eating the hulls. They're eating the bolls. The fall armyworms, we've got cage loopers and bollworms, and once you eat through this, that messes up the entire process of that seed developing, the fruit developing, and the fruit is actually the fiber. It's actually the cotton. And so we can see that over time, the amount of insecticide that we've sprayed has decreased as we've increased the adoption of Bt corn. We might expect this to continue on, but this to now start to make a move back up. This is a little old. This is 2007. We're going to have to probably get some updated, or at least the corn acreage, right? But the insecticide spray might be increasing. So just to give you a basic idea of, like, we've got to breed these plants in order to adapt to the changes in the environment for insects, pests, diseases, drought, salinity. Someone even mentioned that we have excess rain. And so how do we breed for that? What is the gene that is affected by that rainfall? All of these components play a role in why we're going to use these new techniques, CRISPR-Cas9 and QTL, in order to speed up the timeline to get the product to the producer. Because we don't have 8 years to just get the first batch out. We need this in 3 or 4. And that's why Module 2.2 will at least introduce you to some of the techniques that we can use in order to meet these challenges. All right, that's all I got for now. We'll see you in the next video.